Hello and welcome. I'm Gauhar Raza and you are watching Eureka. Tuberculosis is a dreaded word. People are scared of those who have acquired this disease. There's a stigma attached to it. About 1/3 of the population of this globe is affected by tuberculosis, which means any 3 people you see, there is a likelihood that one of them has tuberculosis. But in most cases, in fact, about 90 to 95% cases, it remains dormant. Only 5 to 10 people get affected in a way that it becomes active. About 9 billion people have active tuberculosis, and about 1.5 million people die almost every year. It's a disease today of poor countries, developing countries. If you look at the map of the globe and interpose it with the incidence of tuberculosis, then Africa becomes completely red. India is not far away. Asian countries are intensely affected, but Europe and North America are in a very comfortable position. Very few people get affected by tuberculosis in those developed countries. How does that happen? Why is it that it has been wiped out almost completely from Europe, but developing countries are still affected? We as human beings spend about 20% of our science and technology research budget on armament. If 1% of that budget is spent on research about tuberculosis, probably we will be able to wipe out it from the globe. But that has not happened in so many years. Today, we'll be discussing all these questions. And I have with me here Dr. Shekhar Mande. Very welcome.
Thank you very much.
To Rajya Sabha channel and Eureka.
Thank you very much.
This question has troubled me and many people a lot. That how come in 50s and 60s, tuberculosis was almost to very low level in Europe, but it remains so high as far as incidents are concerned in developing countries. How does that happen?
So the disease itself is a very complex process, and generally transmission from one person to another would have a higher probability in crowded places. So even when it researched in the 80s in the Western countries, it essentially researched in the cities like London or New York and all, where there was higher population density and there was higher chance of bumping into another person who was infected with TB. You see, and we have generally a higher density population in the country than in the Western countries.
But Africa doesn't have high density population, especially southern part of Africa, where the incidences are very, very high. In fact, about 300 per thousand or something is the figure in Africa.
That's right. So TB also has a very strong correlation with certain other kinds of diseases, and the most common which is known is HIV, and the percentage of HIV in the African subcontinent is higher, and therefore co-infection with TB is higher there. In our country, we know that there is correlation between diabetes and TB. That also appears to be one of the factors that could actually explain. But I don't think we really have reasons for that, apart from these many different factors: crowding of the places, co-infection with other disease, and so on and so forth.
Tobacco consumption?
Tobacco consumption also is likely to have a significant correlation to TB.
We are spending a lot of money because it's our disease. Developing countries don't have to bother too much about it. But a country like India cannot afford not to spend money on researching in this particular area. How is it that Europe has been able to wipe it out without the high kind of research, the cutting edge technology that is available today in India and elsewhere also? We are able to not overcome it. Yet, in 50s and 60s, when you didn't have those crystallography machines, they were able to do it.
So, it will not be entirely correct to say that Europe and US have wiped out TB. So, they also continue to have significant incidence of TB. We seem to have bit higher incidence of TB than
Very
Europe.
high as compared to Europe.
Correct. And the reasons are not very easily explainable. As I said, there's very different reasons that we talked of earlier. There's yet another reason that also people talk of is the efficacy of the Bcg vaccine. And it appears that the Bcg vaccine that all of us have taken a shot at is more effective in European or Caucasian population than in the Indian population. A very famous trials called Madras trials, which had actually indicated that Indian population may not be very effectively protected by the BCG. But these are all reasons which all go together, I mean, there's not one or two factors that we can point out that why incidence of TB appears to be higher in our country.
Do you agree with the proposition that countries like India and many countries from Africa, where the incidents are very high, should come together and try to wipe it out?
Absolutely. There's no doubt about it that it's our duty to actually work on TB and find out ways of preventing, curing, and rapid diagnosing the disease. So there are three main arms of containing any disease, and TB being so important, I think it's our collective duty to try to work on all the three aspects.
But Chinese experience also shows that China has been able to successfully reduce the incident levels to a comfortable level.
Well, not really.
Especially in last 10 years.
Yeah, not really. Actually, once again, all the three arms, if effectively implemented, perhaps there is a way of controlling the disease per se.
Did you think that you will be doing science?
Yeah, that I can say more definitively that yes, actually, I had made up my mind quite early on that I would like to do science.
That was a definite thing when you were a school student.
Right. That's it, absolutely.
That you will be doing
absolutely. When all my classmates were going for engineering and medicine, I decided that I would rather go to science then.
And research.
And research, that's right.
And physics.
Physics was the chosen subject then, yes.
Your father was a physicist.
That's right.
I'll take a break here, but the discussion will continue. We'll come back soon. Don't go anywhere. Welcome back. Dr. Shekhar, we were discussing that as a child, your family was a very well-educated family. You don't come from a family where the education was not there. So many families in the country, and they have thrown up very bright scientists. But you right from the beginning had an atmosphere at home where physics was being done and your mother was teaching French. Did it have a pressure on you to excel? Were you ever scolded for bad marks in doing badly in education, or
not
it was
really
a natural thing that you will do good?
Absolutely. There's not really. I don't think my parents put any pressure on me to excel in studies or per se. So things were just happening, and whatever happened, I mean, used to happen. I mean, I don't think I ever got a scolding that why do you get 90 marks and why not 95? That kind of scolding never happened.
Today it happens so often in most of the families.
Absolutely.
That why didn't you get 99%? You will not get admission in this university or that university. When did you decide to do exactly physics, and which area interested you most?
So around the time when I was in 9th or 10th, I was slowly making up what interest would be after 12th. So the 12th was the deciding year, the 12th standard. And by then I had made up my mind that I would rather come into physics as the general training and then take a call at a later stage which sub area of physics we would enter into.
And your father became role model by that time.
That's right. So father being a physicist helped.
Did he discuss physics at home with you?
Yeah, we did actually. Quite a lot of times he used to discuss some of the basic aspects of physics. Even as when we were kids, why is the sky blue or things like that. And why do material left on their own over a period of long time, they would try to form specific assemblies, regular assemblies and things like that. I mean, we used to have general discussions on some aspects of nature.
Dinner table and elsewhere.
Yes.
Did they take you to various museums, etc., and were you exposed systematically to ask questions?
So father had a small collection of crystals. We used to go to the physics department to see once in a while these astronomical events. So they used to have a telescope there. We used to go and see them. In I think around 1980, if I'm not wrong, there was a total solar eclipse, and I went to Khammam to see that. And I was given an experiment to see behavior of cockroaches during the eclipse time. We of course forgot about cockroaches by the time total solar eclipse happened. We were all looking up.
Yeah.
So those are the kind of things which were happening.
So you were looking in the nook and corner of rooms and places, or was it a controlled experiment?
We were given a box of cockroaches and asked to observe them and write down the observation. When total solar eclipse happens, what happens to the cockroaches?
And what was the finding?
So as I said, we forgot about the cockroaches because that site was so spectacular.
Ran away to see the total solar eclipse.
Absolutely, the site was spectacular.
Which was much more probably interesting than cockroaches.
Yes, right. And we were asked to spread this white sheet and see those waves that come before total solar eclipse and things like that.
It's amazing phenomena. I'm sure the earlier human beings must have been enamored by it totally, and they didn't know what is happening to the world and cosmos. Coming to your research, you did excel in physics, but then it started a different field. Somehow you got interested in biology, and then how did you train yourself?
So it actually happened gradually.
For something very new, in fact.
Yeah, but when you do things, you always want to do something new, right? Anything that you do, even in physics, if you start doing condensed matter, if you do quantum, they're all always new. And I guess it happened gradually, because I did my PhD in biophysics. And when I went for postdoc, with a person called Wim Hole, he was working on trypanosoma and malaria and all. So naturally, we started reading about African sleeping sickness and malaria. And when I came back to Chandigarh, then probably by then I had made up my mind.
Why did you come back? You had such a good mentor. He was your role model once again.
That's right.
After your father. So why did you come back?
I think coming back was a natural decision. So doing something back here is always which fires many people up, and that's probably one of them.
Was it cultural difference that you experienced there which bothered you and you wanted to come back?
Could be one of those reasons, but that's not the only reason.
Not because of parents?
Not because of parents, certainly.
Again, parents didn't put any pressure?
Parents didn't put any pressure at all, in fact. So cultural difference is one of the aspects, but something to do for the country and something which is relevant for the country. They're all the different reasons that one can think of.
Why did you get interested in tuberculosis?
So as a postdoc, I was working on leprosy, a protein from Mycobacterium leprae. I was working on that. And then I had started reading about tuberculosis, and the problem of enormity, that what you just now spoke of, that occurred, that it does require actually deeper thinking. So in some way, if you can contribute in a small way towards understanding the disease, that would be useful, is what I thought.
But for a long time, you were also working with DNA Fingerprinting Institute.
Yes, but DNA Fingerprinting Institute...
It was incidental, or you were interested in DNA fingerprinting?
No, I must say, I must confess, and with apologies to the directors of DNA Fingerprinting Institute, that the name Center for DNA Fingerprinting and Diagnostics is a bit of a misnomer, because the institute started with the objective of DNA fingerprinting.
Yes.
But when Dr. Hasnain was the director, he decided that he would grow up many different research groups, which would eventually augment the capability of diagnostics and DNA fingerprinting. So we were actually doing kind of science that we were doing, with the hope that it would be useful for...
There also you were working on tuberculosis.
That's right.
It's the same area. You never left once you got into
That's right.
tuberculosis.
That's right.
That has been your specialization throughout.
That's right.
I have to take a break. Don't go anywhere. We'll come back. Welcome back. Dr. Shekhar, we were trying to understand that how your interest shifted to biology gradually, from physics to biophysics, and then deep into cell and at molecular level and crystallography. Did physics help you, training in physics help you to understand the phenomena in cells and tuberculosis and bacterium? Did it help you?
It does indeed help. So, the
Or it was a complete departure?
No, it's not a complete departure in any case. Even today, I have a project which is going on in the lab, which tries to understand very large scale networks. This is not crystallography, and it has a lot of aspects of physics. But what physics essentially helps you in the training is to look at things very objectively and in a more quantitative fashion. While I guess most of the biologists are trained the manner that is not a very quantitative kind of a thinking. I mean, that's my subjective opinion. But physicists do actually try to look at quantitative aspects into biology. If a physicist who moves into biology always tries to bring some quantitative aspects, and there some thinking, actually different kind of thinking does help.
And this is the period when the straight jacketed disciplines and very, very tight walls between disciplines were crumbling down.
Absolutely. I think
that offered you an opportunity.
Absolutely. I think very well said by you that even in our own country, we have large number of examples who are outstanding biologists today in the country. Some of the topmost names actually came from a completely different background, either physics or chemistry. So breaking the walls between different subjects has been a very phenomenal thing in our lifetimes.
It's been a revolution in science.
Yeah, I guess it probably started 50 years ago before we did, because there are a number of examples before us. In fact, I mean, a lot of examples were inspired by people like Max Delbrück, for example. Right? So Max Delbrück's initial hypothesis on to find genes, for example, in using Drosophila as a model. So physicists are also used to model systems and all, and Delbrück brought the concept of model systems in biology. That inspired eventually Schrödinger to write a book called What is Life? And many people are on record saying that they were inspired into biology after reading Schrödinger's book. For example, Francis Crick is one of those.
One of the biggest names in physics.
That's right.
Who influences biology.
That's right. And I think the thinking must have started 50 years before us, but that does actually help to have those kind of.
You got Bhatnagar Award, which is one of the most coveted awards in the country. What did you get it for?
So, so Bhatnagar Award is a recognition of the overall work that you do, but the citation essentially said that my work on a class of proteins called chaperonins. These are a class of proteins which help other proteins in the cell to fold. They're also called heat shock proteins, and a nomenclature goes by their molecular weight. HSP 60 is 60 kilodalton molecular weight,
right.
HSP 70 is 70 kilodalton molecular weight, things like that. So work on those is what we have done in our lab.
What was exactly that you were trying to find?
So we had been interested in HSP 60 class of chaperons, and when we started working on this class of chaperons in Mycobacterium tuberculosis, there's the bug that causes TB, we realized very soon that it doesn't have the canonical properties what it should have. For example, for a chaperon to bind a substrate and fold that substrate into native form, it has to form large ring, and the ring is of 14 subunits. So 14 equal kind of proteins, they together form 7 and 7 back to back. We realized that the Mycobacterium tuberculosis chaperon or HSP 60 does not form that kind of a ring. So that actually was a significant deviation from the textbook knowledge that we had learned about chaperons.
Or what other groups were doing across the world.
Correct.
But this understanding, does it have practical implication?
Yes, it does have, because then we actually try to understand then that what role, potential biological role it has in TB, and maybe it has a different role than folding proteins alone. And if that does have, then maybe we can actually target this protein for a useful purpose later.
You've published about 100 papers in some of the best journals across the world. You are member of so many scientific organizations. You've got best of the awards. So this is recognition. Now I'm going to ask a difficult question, but this question I've been asking to everybody. It's very difficult to say that this paper of mine or this work that I did was, or this recognition I value the most. But let me ask you, was there a moment of Eureka when you said, yes, I have hit the ball?
Yeah, admittedly, you also admitted it's a very difficult question. The Eureka moments actually happen very less frequently in anybody's careers, right? But there are certain times that you get very excited about your new finding, for example. So one of those days when I was doing PhD and we were working on very challenging problem of solving the crystal structure of a protein called peanut lectin, that would be the first structure determined in the country. And Professor Vijayan was really the driving force behind that. And he had taken that ambition despite criticism of so many of his peers. And he was determined to show that he can do it. And we all became a party to that, right? I mean, his awe and...
And when you did it, the whole team
That's right. So
celebrated it?
So the day we calculated what is called as the electron density map, and we were looking at it, and then we knew that we have essentially cracked the structure, I think that was a very exciting moment. And Professor Vijayan himself was extremely excited.
How did you celebrate it?
I don't remember, it was 1991, but it is a great moment for all
Because
of us.
generally the image about scientists is that they are very dry people, they don't celebrate, they don't get excited easily, they are very skeptical about everything.
That's right. So that was a great moment. I mean, I would say that during
You
PhD,
still remember it.
I still remember
This is
it.
long back.
That's right. And everyone around was very excited about Professor Vijayan, his very close collaborator, Professor Sorolia. Then we had another very good mentor, Professor M.R.N. Murthy. All of them were very excited because we are actually seeing the protein structure being solved in the country.
Now, again, let me change the subject. Would you argue that a poor country like India spends so much of money on scientific research? Some people may argue that let's do what we can do, which is jugaad, and let others spend money on research. So, as a scientist and as head of the institution that you are working for, would you argue that more money should be spent on scientific research?
Absolutely. I mean, I would strongly argue against what you said, that let others spend and we'll do jugaad. That's not really the philosophy. Our population and our problems and our society are actually our problems to solve. I don't think anyone else is going to come to our country and say, solve your problem.
Will you agree with me if I say that if we give up science, then we jeopardize our future?
Absolutely. There's no doubt about it. There's no doubt in my mind that if we don't increase our budget on science, we're actually jeopardizing our future. See, we have so many problems like malnutrition is a raging problem in our country. Number of children dying below the age of 5 of many different diseases, including malnutrition or preterm birth, right? We have to find solutions to them. Nobody else is going to come and say, this is a solution for you. This is a magic bullet for you.
Are you arguing that we should be spending money only on these problems and not on space research?
No, I'm not saying that. Even space research has actually benefited the country enormously, right? So the kind of money that we are spending in 80s and 90s or even today, as you see the benefits that you see today of space research, what has happened. So information technology revolution that we have gone through has direct bearing on the money that we spend on space research. So spending money on all aspects of science and the technology that comes out of it and its implementation for the benefit of the society are all important aspects. And we must not therefore withdraw from spending money on science.
If we neglect one area, we are essentially neglecting other areas as well.
Absolutely.
Because science is an integrated whole.
Absolutely. And I would argue rather for increasing the funding of science to at least about 10 times than what we have right now.
Which China has already done. We haven't done it. Our expenditure on scientific research has been stagnant. Anyway, at the end of the program, I ask normally the question that what would be your message to the younger generation?
I think very difficult to give messages. I mean, I'm not used to giving messages like this. But I would just say that follow the pursuits that interest you the most. There are lots of questions around nature, about nature, that are very exciting. We still don't understand the nature completely in its entirety, and I don't think we would ever in the next maybe a few hundred years. But the questions are so exciting that younger generation can actually start contributing to trying to understand some fascinating aspects of nature.
Ask questions and try to answer them. May I, on your behalf, promise our viewers that you will be happy to answer questions if they have?
Sure.
Write to us. Our email address is eurekarstv@gmail.com. We'll be back next week with again a fascinating scientist, an outstanding scientist. Till then, goodbye. Thank you very much. It was just wonderful talking to you.
Thank you. My pleasure.
It was simply great.